Hi everyone. Here we are discussing about the bacteria and the fungi. So how we will identify the bacteria and the fungi in laboratory on the basis of their uh, molecular characteristics. For bacteria, if we want to identify in the lab, so we can target the cystine rRNA gene. So for this we can use the 26F and forward primer and 1492 reverse primer. So the size of the amplicon is about 1540 base pair. Whereas in case of fungi, generally people target internal transcribed spacer region. This is very commonly known as ITS region, and its size depends on which primer we are using in the laboratory or which region we are targeting. So for example, if you are targeting ITS1 and ITS4 primer using this primer, so then the length should be approximately 700 base pair. So it is very helpful to know which type of pure culture we have in the laboratory, or if suppose for example we don't know, we are isolating a new culture, also we can identify the community from the metagenome for both, for fungi and the bacteria, and we can characterize the bacteria up to species level. Also we can characterize the fungi up to species level. However, there is some drawback of these methods, both methods. So the drawback is what is that drawback? Because both of these methods have limitation, and they cannot identify, they cannot differentiate some of the species due to, for example, this is species A and this is species B, both share the same cystine R, both share the same cystine R RNA gene or ITS gene for fungi. So if the A, for example, A is fungi, it show the similar ITS, so we cannot differentiate them. And also here as well, A is bacteria, B is bacteria. If they share same cystine, then we cannot differentiate them. So this is the limitation of these methods. In such cases, we will target more another molecular marker that can differentiate them